Presenting data from a recently completed prospective phase 2 window of opportunity clinical trial. Tamoxifen, which is the widely used drug in breast cancer, how it impacts gene expression when this drug is given to patients who have never been treated, who did not see any therapy, who never had previous cancer or anything like that. This has not been done from this angle, so this will be the first study to look at that gene expression. So what we have done is we give tamoxifen, randomly distribute patients into two groups. One group will go through usually the standard of care, which means they wouldn't usually get any medicine until they go through surgery for 1 month later. The other group, we give tamoxifen therapy for 4 weeks. And then after 4 weeks, the tumor is resected as usual. And then from that tumor, we do all various analysis, and including very sophisticated high resolution gene expression analysis called RNA-seq. So that's the data which I'm going to present. We recruited a total 59 patients between Roswell Park and my collaborators institute, University of Chicago Medicine. And of the 59, 52 patients completed the study, and those are the samples we analyzed. Tamoxifen, and its conventional role of tamoxifen has been that it will bind to estrogen receptor and prevent estrogen from binding, because estrogen is the hormone which activates the receptor, downstream activating a whole bunch of genes, whereas tamoxifen will compete it, so it's called a competitive inhibitor, and bind it to estrogen receptor, thereby inactivating it. That's known for years from various research groups. What we found was that in addition to targeting the conventional targets, estrogen receptor can also target genes regulated by tumor suppressor protein P53. P53 is one of the most important tumor suppressor protein, which is inactivated one way or the other in most cancers, and breast cancer is one of those. In this particular cancer, a subtype of breast cancer we looked at is called luminal breast cancer. There, P53 is genetically normal. In scientific terms, it is called wild type. However, it is functionally, basically it is dysfunctional. Even though, so that's where estrogen receptor comes. So estrogen receptor comes and binds to the P53 protein and inactivates it. So this we knew several years ago. We published this few years ago, both in the cell models and in animal models, and also in a retrospective clinical trial in collaboration with a group in Germany and Baylor College of Medicine. So, but we did not, that was consistent, all the data, but it still did not prove that this interaction indeed occurs in patients, and when you give tamoxifen, this interaction is disrupted. So that's what we showed here. And as a result of that disruption, whole bunch of hundreds of genes, more than 400 plus genes, are differentially up or down regulated in tamoxifen treated patients compared to the untreated patients. So the implications are big in the sense that right now, even though P53 has been known to be a major tumor suppressor, especially in breast cancer, there is no, P53 is not factored into decision making of therapy, where for various reasons, because directly targeting P53 has been tough, and some of the past data on P53 because of the reagents issue has been equivocal, but now we have far better resolution techniques. So this would imply that, our data would imply that depending on the P53 status, the tumors can respond or not respond to tamoxifen. So that's the big thing, because even though theoretically all ER positive breast cancers are supposed to respond to tamoxifen because the drug is against the receptor and receptor is there, but in real life one of the main problem in the clinic is that many such patients don't respond to tamoxifen to begin with. Even if they respond, after a while they develop resistance. So our finding says that it is one of the important mechanisms behind that is that p53 status is different. If p53 is so-called normal p53, it will work, the patients with such tumors will be responsible. But if the patient's tumors have mutant p53, then even if tamoxifen disrupts the ER p53 interaction, the resulting p53 is genetically dysfunctional to begin with, so therapy will not be working. That's the implication. So to put it another way, our data will enable, we have to go through a larger clinical trial, of course, enable to make a decision which patients will be receptive or which patients will be benefited by tamoxifen therapy and which patients will not be. Because right now, any woman who walks into the clinic at a breast cancer, first thing clinicians do is to check for estrogen receptor. As far as they are estrogen receptor positive, invariably they get tamoxifen therapy, but many of them don't. It won't be beneficial. So we will be able to stratify the patients based on p53 status, and that will have a major implication in conventional therapy. And another thing which we did not address in this trial, because the trial is always limited, because unlike other animals or cells, it's hugely time consuming and hugely expensive, so you can only focus on one small thing in a clinical trial. But what, in combination with this from other studies, what we see is that even in cancer subcategory of breast cancer called triple negative, where the receptor is not there, we see that tamoxifen can be beneficial if we stratify those patients in terms of p53 status. So that's something new, but that is yet to go through a clinical trial side. But this is what we already know in patients. So it is very gratifying to see whatever we saw in the cells and animals, now we can see in real patients. So that's the big strength of the clinical trial.